Imagine a factory producing life-saving medicines. These medicines, injected directly into our bodies, must be completely sterile. This means absolutely no bacteria or other microbes can be present. But how do we guarantee this level of cleanliness? This is where media fill validation comes in. Media fill validation simulates the actual drug manufacturing process. However, instead of the real drug, we use a nutrient-rich broth called culture media. This broth acts like a feast for any hidden microbes. If the broth remains clear after incubation, it indicates that the process successfully eliminates contamination. A cloudy broth, on the other hand, spells trouble, signifying microbial growth and a breach in sterility. Media fill validation is not just a regulatory checkbox, it's a critical safeguard for patient safety. Injections bypass the body's natural defenses, making contamination extremely dangerous. Contaminated medicines can lead to serious infections, even death. The consequences of a single failed batch can be devastating, both for patients and the pharmaceutical company. Media fill validation provides assurance to both manufacturers and regulatory bodies. It demonstrates that the manufacturing process consistently delivers sterile products, protecting patients and maintaining public trust. There are three main types of media fill validation: startup, ongoing, and requalification. Each serves a specific purpose in maintaining sterility throughout a product's life cycle. Startup media fill, as the name suggests, is performed before a new production line or product is released. It validates the initial sterility of the process. Ongoing media fill is conducted routinely during production, ensuring that sterility is maintained over time. Finally, requalification media fill is triggered by specific events, such as changes in equipment, processes, or personnel. It revalidates the process to ensure that any modifications haven't compromised sterility. Media fill validation is a resource-intensive process, requiring specialized equipment, materials, and trained personnel. Key equipment includes autoclaves for sterilization, laminar flow hoods to provide a sterile working environment, and incubators to grow any potential microbes. The culture media itself is a critical component, carefully selected to support the growth of a wide range of microbes. Trained microbiologists are essential for designing and executing the validation study, analyzing the results, and providing expert interpretation. Collaboration between quality assurance, manufacturing, and microbiology departments is crucial for a successful media fill validation. Section 5. Step-by-step process of media fill validation. The media fill validation process involves a series of carefully orchestrated steps. First, the media is prepared and sterilized, mimicking the actual drug preparation process. Next, the sterilized media is filled into containers, simulating the filling process of the final product. The filled containers are then incubated at a specific temperature for a predetermined time, allowing any potential microbes to grow. During incubation, the containers are closely monitored for any signs of turbidity, indicating microbial growth. Finally, the results are carefully documented and analyzed. If no growth is observed, the process is considered validated, demonstrating its ability to produce sterile products. Section 6. International Guidelines and Standards. Media fill validation is not a haphazard process. It is governed by strict international guidelines and standards. These guidelines, established by organizations like the US Food and Drug Administration, FDA, and the European Medicines Agency, EMA, provide a framework for conducting and documenting media fill studies. They cover aspects such as the type and number of units to be filled, incubation conditions, and acceptance criteria. Adhering to these guidelines ensures that media fill studies are scientifically sound and their results are reliable and internationally recognized. This harmonization is crucial for ensuring the safety and efficacy of pharmaceutical products across the globe. Section 7. Evaluating and measuring media fill results. The success of media fill validation hinges on the accurate evaluation and measurement of results. The primary indicator of contamination is turbidity, a cloudiness in the media-filled containers. This cloudiness is a visual cue of microbial growth, indicating a breach in sterility. Microbiologists meticulously inspect each container, carefully documenting any signs of growth. In cases of suspected contamination, further tests are conducted to identify the specific microbe present. This thorough analysis helps pinpoint the source of contamination and allows for corrective actions to be taken. The results are then compiled into a comprehensive report, providing a detailed account of the media fill study. Section 8. Documentation and revalidation in media fill. Documentation is paramount in media fill validation. Every step from media preparation to result interpretation is meticulously recorded. This documentation serves as an audit trail, demonstrating compliance with regulatory requirements and providing evidence of a robust sterility assurance program. In the unfortunate event of a media fill failure, thorough documentation is crucial for investigation. It helps identify the root cause of the failure, guiding corrective and preventive actions to prevent recurrence. Revalidation is then performed after implementing corrective actions to confirm their effectiveness and ensure the process is back in a state of control. Section 9. Importance of collaboration in media fill validation. Media fill validation is not a solo act. It requires seamless collaboration between different departments within a pharmaceutical company. The microbiology department plays a central role, designing the study, analyzing the results, and providing expert interpretation. The manufacturing department is responsible for executing the media fill process, ensuring it accurately reflects the actual drug manufacturing process. Quality assurance oversees the entire process, ensuring adherence to protocols and regulatory standards. This collaborative effort ensures that the media fill study is comprehensive, accurate, and truly reflects the sterility assurance of the manufacturing process. Section 10. Real-world examples of media fill success and failure. The importance of media fill validation is best illustrated by real-world examples. A successful media fill program provides confidence in the sterility of manufactured medicines, protecting patients, and upholding public health. On the other hand, failures in media fill can have significant consequences. One notable example is a case where a contaminated batch of intravenous drugs led to a widespread outbreak of infections, resulting in numerous hospitalizations and even fatalities. This tragic incident underscores the critical role of media fill validation in preventing such disasters and safeguarding patient safety. Section 11. Conclusion: the enduring importance of media fill validation. In the realm of pharmaceutical manufacturing, sterility is non-negotiable. Media fill validation, though a complex and resource-intensive process, is an indispensable tool for ensuring the sterility of injectable medicines. It provides assurance to manufacturers, regulatory bodies, and most importantly, to patients, that the medicines they rely on are safe and free from harmful contaminants. As technology advances and regulations evolve, media fill validation will continue to adapt, ensuring the ongoing safety and efficacy of the life-saving medicines we depend upon.